Thank you and good morning, everyone. It's great to be here. It's great to see everyone come out for this inaugural event. Let me just start. I need to start by extending a warm welcome to everyone and also to thanking the members of the Dartmouth International Vaccine Institute, who are the primary organizers of this. And Ford showed you their pictures. I'm going to make sure you hear all their names. So Ford von Rhein, Kendall Hoyt, Lisa Adams, David Lieb, Peter Wright, Omar Sajad, Margie Ackerman, David Katz, Lindsay Leininger, Cody Meissner, John Modlin, Patricia Monseri, and Brendan Nyan. They are a cross-campus group of people who have come together to help put this together for us today. So I would like to thank them publicly for taking that initiative. I'm also going to reiterate our thanks to the sponsors. These events don't happen by themselves, so we've had really generous support from the Jack and Dorothy Byrne Foundation, the Dartmouth International Vaccine Institute, the Dartmouth John Sloan Dickey Center for International Understanding, the Department of Microbiology and Immunology at Geisel, the Dartmouth Center for Global Health Equity at Geisel, and the Tuck School of Business Center for Healthcare, and finally Dartmouth Health Department of Pediatrics and Infectious Disease. So I want to thank everyone who helped organize this and everyone who's helped sponsoring this. It's been a really important cross-campus effort. So now it is my pleasure to introduce our speaker for the inaugural Samuel L. Katz Memorial Lecture, which is the keynote address for our event today. And that's Dr. Jason McClellan. Dr. McClellan is a professor in the Robert A. Welch Chair in Chemistry at the University of Texas. He's developed an exceptionally powerful research program, really a tour de force, if you will, focused on elucidating the molecular mechanisms of host pathogen interactions, particularly those that involve viral glycoproteins and viral host cell interactions. His group determines protein and glycoprotein structures and develops tools needed to answer basic science questions that are readily translated into therapeutic interventions such as vaccines. Over the years, his work has brought insights into the respiratory syncytial virus, C. difficile, Crimean Congo hemorrhagic fever virus, and most notably coronavirus, including MERS and SARS-CoV-2. His seminal work on the coronavirus spike protein that he conducted as a faculty member in our Department of Biochemistry and Cell Biology serves as the basis for all of the currently used vaccines in the US and Europe. It may also be being used in other parts of the world. It's just not entirely clear. They're not disclosing that to us. It's probably not an exaggeration to say that we may still be searching for a solution to the SARS-CoV-2 pandemic without his discovery. In recognition for his scientific discoveries in protein structure and how they translate to real-world human impact, he's received numerous awards. That includes the 2020 Golden Goose Award from the American Association for Advancement of Science, the 2020 William Prusoff Memorial Award from the International Society for Antiviral Research, 2019 Viruses Young Investigator and Virology Prize, the 2018 American Crystallographic Association Editor Early Career Award, the 2015 Charles Hood Foundation Child Health Research Award, and more recently, the National Academy of Sciences Award in Molecular Biology, the Welch Foundation's Norman Hackerman Award in Chemical Research, and in 2022, he was the inaugural recipient of the McGuire Family Prize for Societal Impact at Dartmouth. I'm delighted to ask Dr. McClellan to come to the podium, and please join me in welcoming Dr. McClellan back to Dartmouth.
All right. Well, thanks so much, everybody, for coming out. So it's very exciting to be back here. Thanks, Dwayne, for that very warm introduction. I'm really excited to be the inaugural presenter for the Katz lecture. And I'm going to tell you all about some of the work we did here at Dartmouth on coronaviruses, and then that we could continue to do in Texas. All right, I do have conflicts of interest, as you know, and I have to show this. So we are inventor on patents related to prefusion stabilized coronavirus spikes, their use as vaccine antigens, and on coronavirus neutralizing antibodies, which will be relevant. All right, so for vaccine development, we have to make a number of decisions about the types of vaccine we're going to create. Some of these don't require much in the way of atomic level information about the pathogen, right? Like we can make inactivated or live attenuated that just use, in this case, the entire virus. And those have worked, maybe more for some of the traditional vaccines, measles, polio, and others. And then there's a variety of different vaccine modalities where we're going to deliver one or more individual antigens from that virus, right? And so we might just express and purify some recombinant form of a viral protein. So in this case, if this is coronavirus and that spike, maybe we deliver full spike, some truncated portion, like just the receptor binding domain. We could, rather than expressing and purifying that antigen itself and injecting it, we might ask the body to do the expression. So in that case, the antigen would be encoded by the DNA, the mRNA, or virus vector. We can make VLPs that display one or more antigens. And so for these approaches, which we're most interested in, we really have to figure out what's the optimal form of the antigen to present to the immune system. And that's what my lab tries to work on, using structural information about the protein, its interaction with the human immune system, and how to generate the ideal antigen to elicit the best possible immune response. One thing I like to show is that we've known about coronaviruses since the 1930s, right? At least the general public thinks we've only heard about them for the last couple of years. But like a lot of good science, it started with some of the veterinarian and animal work. So this dates back to 1931. This is the first coronavirus discovered, infectious bronchitis virus, so an apparently new respiratory disease of baby chicks. So since 1931, scientists have been studying coronaviruses. The first human coronaviruses were discovered and isolated in the 1960s. So again, quite a bit of research on those. Relatively small field. SARS helped put it on the map and generate more funding and interest. So we had the SARS outbreak in 2002. That led to the discovery of two more human coronaviruses. NL63 and HK1. And so these two and those two are causes of the common cold. So many of us have been infected by one or more of those. Then there was the second zoonotic event, the MERS coronavirus, which spilled over in Saudi Arabia and the Middle East in 2012. That was one of the reasons Barney and I started working on coronaviruses, because we now had two zoonotic events a decade apart, and it seemed like maybe they were going to keep happening, and that wasn't going to be the last one. And then, of course, the SARS-CoV-2 pandemic in late 2019. These are a very large family of enveloped RNA viruses. So this shows there are 4 different genera. There's alpha coronaviruses, of which 2 of the endemic coronaviruses are part, 229E and NL63. And then the beta coronavirus, that's the largest genus with different sort of lineages, lineage A, B, C, and D. So lineage A has 2 of the more common cold viruses, HKU1 and OC43. And then lineages B and C have the zoonotic, the SARS-like and MERS-like. And then there's also some animal back coronaviruses in the Gamma and Delta. And this is important for when we think about trying to generate broad universal coronavirus vaccines or pan-beta coronavirus vaccines. Like, how broad can we get? Right now we're sort of having trouble making one vaccine that just covers SARS-CoV-2, right? So then you might step out and think, can we get something that covers all of SARS or SARS and MERS? Can we make a pan-beta or truly universal? But that type of diversity makes it difficult for vaccine development. I think as many people know, the surface of coronavirus is decorated with spike proteins, and that's been appreciated since really they were discovered in the 1960s, the human coronaviruses, and people looked at them using electron microscopy. So you can see that they have these little club-like projections. This was back in 1967. So this group of researchers in Nature in 1968 put forward the name coronaviruses. They note that the particles are more or less rounded in profile, so not perfectly spherical, somewhat pleomorphic, but they have this characteristic fringe of projections, and its appearance recalls the solar corona with its projections. So said, let's call this family of viruses coronaviruses, and that name has stuck. This was our plan. we did a lot on structure-based vaccine design when I was a postdoc at the Vaccine Research Center working with Barney on respiratory syncytial virus. And Barney's going to end the session today talking about the development of the RSV vaccines. So we're kind of doing it in an opposite order, but the plan was to do everything we did for RSV and apply it to coronaviruses. So when I left the VRC to come to Dartmouth in 2013, this was our plan. We needed structural information. Right, so this was how, even in like 2016, people depicted coronavirus spikes, and that's not high enough resolution to do structure-based vaccine design. We had to determine a much higher resolution spike structure, and then using the structure of spike, start performing the protein engineering to try to make the optimal antigen that would express well, that was the most stable, that would elicit the most robust immune response, and then obviously test that. And so this was work we've been doing with Barney since then. As I mentioned, even at this review, early 2016, we still didn't know what the true coronavirus spike looked like on the surface of the cell, on the surface of the virus. And we had images of individual portions, right? So this is a really large protein, generally around 1,200 to 1,300 amino acids, and it's a trimer. So 3 copies of it come together to form the full spike. And so X-ray crystallography was really helpful in solving structures of individual fragments. So we knew that the S1 subunit is involved in receptor binding. It has an N-terminal domain that has a galectin-like fold that can often bind sugars and help just some weak affinity, weak attachment to the cell. And then there is a larger C-terminal domain in S1, generally referred to as the receptor binding domain, or RBD. And that has a conserved motif, this beta sheet structure, and then it has variable loops emanating out. And the variable loops are what interact with receptors. And so there have been structures of those determined alone, the RBDs alone, and then complex with their receptors. What I find really interesting is that the receptors are always peptidases, but the peptidase activity isn't required for entry. So some of the receptors like ACE2, aminopeptidase N, dipeptyl peptidase 4, the viruses keep evolving to recognize different peptidases, but that activity isn't required. So maybe something about the shape, height off the membrane, clustering, expression, but it keeps evolving to recognize peptidases. So we had some information on these 2 domains in S1. S2, which is the fusion machinery. This is part of some spring-loaded machinery that's needed to fuse the viral membrane in the host cell membrane. We only had some crystal structures of 2 parts, this 2 heptad repeats that form a highly stable 6 helix bundle in what we call the post-fusion conformation, the conformation that occurs after binding. For studies on the whole spike, there were some negative stain EM from Steve Harrison's group and Fong Li on SARS-CoV. So they kind of had, you could see 3 prongs sort of sticking up, and that's really led to that. So we needed to come up with higher resolution information. We started working on MERS because that was what had just emerged in the Middle East. Causes a really scary infection. Like 40% or so of people that get infected die. Fortunately, it doesn't spread as easily from person to person. But we really weren't able to make much of the MERS spike protein. So we could make some individual domains. So we could make the N-terminal domain on S1. We could even make we could make some of the full S1 subunit. But when we tried to express the full S ectodomain that contains the S1, S2 subunit, there's just nothing there. And so that was difficult. Fortunately, Barney's lab had started working on HKU1. One of his postdocs, researchers, went to the Middle East and got a respiratory infection, and we were all really excited that it was MERS. It turned out not to be MERS, it was HKU1, but so we thought, well, we can at least get some antibodies against HKU1. So Barney's group started making the spike proteins for HKU1, and it turned out that these spike proteins are really well behaved. So they could be expressed and purified. We could modify cleavage sites or not. So in this case, we could get nice S1 and S2. We could delete the furan cleavage site and just get a single band for the full spike. We could add a trimerization motif at the bottom to replace the membrane. And these all look really good. So these were nice for structural studies. Unfortunately, they didn't crystallize, in part because of some conformational heterogeneity. They're also highly glycosylated. They're just covered in sugars, which aren't shown here. So we started working with Andrew Ward at the Scripps Research Institute, who's an expert in cryo-electron microscopy. And so working with Andrew's group, they were able to determine the 4-angstrom cryo-EM structure of the HKU1 protein in the pre-fusion conformation. And so we can see one of the S1 subunits is in blue, the S2 subunit is in pink, the other protomers are in gray and white. And this revealed a lot of really interesting structure function in biology. The N-terminal domains were located on the periphery. The receptor binding domains mediated the trimerization at the apex, which suggested that receptor binding could cause destabilization of the spike. It sort of nicely intercalates down and then back up. We identified 2 additional smaller domains, we call subdomains, subdomain 1 and subdomain 2. And then most importantly, it gave us the structure of the spring-loaded pre-fusion conformation of the S2 subunit, which is where we really wanted to focus our engineering efforts on. Okay, so I've been talking about some of the conformational changes, and this will hopefully make it a lot more clear. This is a movie from Digizyme that's going to show spike-mediated entry. All right, so we have viral membrane, one spike, the S2 domain, S1 domain. S1 is sort of a fusion suppressive cap, like the cap of a mushroom that sits on the spring-loaded machinery. Receptor binding causes destabilization and shedding of S1. You then get this refolding of the central helix that harpoons the fusion peptide into the host cell membrane. The pre-hairpin intermediates then begin to collapse as the heptad repeats, form these long coiled coils, and eventually the 6-helix bundle. It just kind of zippers up and brings the viral membrane together with the host cell membrane. So these are really impressive little molecular machines that have evolved specifically to fuse viral membrane with host cell membrane. And a lot of times when you express these proteins, they end up adopting the post-fusion conformation. So the pre-fusion conformation is metastable. It's not the lowest energy form. It's kinetically trapped though. But when you work with it, like we saw Barney talk about for RSV, when you purify it, you generally just get post-fusion. And so we need to use the structural information to design mutations to help stabilize the protein in the metastable pre-fusion conformation to use as the vaccine antigen, because that's what's on the surface of the virus. All right, so this shows a comparison of pre-fusion and post-fusion for a coronavirus spike protein. There is no S1 subunit in the post-fusion because that sheds off. This is very similar to the RSVF protein. RSVF is basically just the S2 subunit. It has no fusion suppressive cap. We really don't understand triggering for RSVF. All of these are class 1 viral fusion proteins, and they all have evolved from one common ancestral fusogen. So you can start to see that there are some elements that are the same. For instance, they all have a central helix in blue. It's smaller in RSVF. The central helix does not undergo much of a conformational change between the two. Right? So the central helix is here and here in pre and post. It's here and here, pre and post for coronaviruses. And then you have these elements that make up the first heptad repeat in green, yellow, orange. All of that is what it flips around 180 degrees and sort of polymerizes up from the central helix. So you get really interesting refolding events. There's coil regions that go to helical transition, and it just polymerizes the helix. Similar here too for RSV, where you have even beta hairpin. So you have some beta hairpin that refolds into alpha helix. So we wanted to use some of the approaches to stabilize RSV to try to stabilize coronavirus spikes. It was a little tricky though, because what we had was the structure of the HKU1 spike, and we were trying to engineer the MERS spike, and those only have about 35% sequence identity. So trying to use one model to design things like disulfide bonds wasn't working. The structures just weren't close enough. We have to be within an angstrom or two to get disulfides to work. One of the tricks we liked from RSV, from my colleagues, Johannes Langendijk, that their method for stabilizing RSV was to put one or more prolines into this hinge region between the central helix that doesn't move and these elements that all need to refold and polymerize the central helix. And so we call this proline scanning, right? This region happens to be among the most conserved regions of beta coronavirus spike. So these are 3 beta coronavirus spikes from the different lineages, A, C, and D. And you can see that a lot of it is different, but this region, they all have RLD, 2 non-conserved amino acids, a conserved glutamate, and then a lot of the central helix is conserved. So my postdoc, Nian-Shuang Guang, here at Dartmouth, started replacing these individual amino acids to proline. Right? So we were trying to do that. And what we can see is that the wild type MERS spike, because I showed you we could barely make any of it, but there's a faint band for wild type MERS. But just changing 1 amino acid to proline, and something that's almost 1,300 amino acids long, gives like a 20-fold boost in protein expression. And generally, as we stabilize class 1 viral fusion proteins, we also see a boost in protein expression. So he changed some of the other ones in this region. We all got, those all led to a nice boost. And the one we liked the most had these 2 amino acids change to proline. And so we call that the 2P construct, and it's about a 50-fold increase in protein expression. And it looked really great. By size exclusion chromatography, we can see this is from the same protein from the same amount of culture. So wild type spike, there's a tiny little peak down here you can barely see, but with just the 2 proline change, huge boost in expression. When we sent these to Andrew Ward's lab to look at them by electron microscopy, we can see that the wild type spike was this mixture of post-fusion, boxed in red, and pre-fusion. And over time, more of the pre-fusion just converts to post-fusion because it is metastable. It wants to go to the lowest energy post-fusion conformation. But with the 2 prolines, it was just all pre-fusion, which looked really nice. So we sent these proteins to Barney's lab, and his postdoc, Kizmekia Corbett, and her team immunized mice. And so this was a pretty low dose, 0.1 microgram dose, which is often needed to tease apart some of these differences. And then they performed neutralization assays. And so higher is better. So we're looking for the elicitation of higher titers of neutralizing antibodies. These were against different variants of MERS-CoV. And they were comparing the 2 proline spike in red to wild type spike in blue, and then just S1 subunit in orange. What we can see is that for many of these, we would get a 5 to 10-fold increase using the 2 proline spike compared to wild type or S1. So this appeared to be a superior immunogen to just using unstabilized spike or an individual subunit like S1. What was great is that because the region is so well conserved among beta coronaviruses, just based on the sequence alignment, we knew exactly where to put prolines into all the other beta coronaviruses. And so we did this for SARS, the original SARS classic back in 2002. Wild type in red, we could make a little bit of, right? But with 2 proline, again, a nice boost. Wild type SARS was also this heterogeneous mixture of post-fusion and pre-fusion spike, whereas 2 proline SARS was all pre-fusion. Even HK1, that we used for the original structure, that the wild type behaves pretty well. We were able to get also a nice boost in protein expression and stability using 2 proline. It worked for OC43. We even cloned and expressed and purified a spike from a bat coronavirus that has not yet emerged into the human population and showed that it worked really well for WIV1. And so we had all of this work sort of patented and published back in, I think, 2017. It got rejected from 5 different journals, which was pretty rough, but that's how it goes. It's been cited more now, so that's good. But it was disappointing at the time because we thought this was a sort of a universal approach for stabilizing beta coronaviruses, and maybe people weren't ready for that yet. But it turned out to be relevant, of course, in late 2019 when everybody's sort of following along on the news and the virologists on Twitter. There was these unknown pneumonia clusters in Wuhan, and so immediately started to think that maybe it's generally either influenza or a coronavirus. In early January, Chinese investigators had isolated the beta coronavirus and had sequenced the entire genome. Barney called me on January 6th to put together a team to start trying to make the world's fastest vaccine. And I said, sure, let's keep doing it. And our plan was to try to stabilize it and get the structure of the stabilized spike. And he was going to be working with Moderna to try to get the mRNA and get all that ready. On January 10th, which was a Friday in the US on the East Coast, the novel coronavirus genome sequence was just released in an online database. Within a couple of hours, Nian-Shuang and Daniel had just figured out where to put the prolines based on the sequence alignment. Within 10 days, Nianchuang had made like 8 different plasmids encoding various forms of the 2-proline stabilized spike. 10 days after that, Daniel expressed and purified the spike protein, started cryo-EM data collection, and by February 10th, we had a manuscript submitted to the Science and Bioarchive describing the cryo-EM structure of the SARS-CoV-2 spike protein. So we went from novel genome to submitted manuscript in 1 month, which was something we're never going to do again. But it was good to do it once and show the students what's possible when properly motivated. So for the SARS-CoV-2, the ectodomain with the double proline, we were only getting about 1 mg per liter, which we weren't super thrilled with. The MERS 2 proline was around 20, SARS was around 10, so this was quite a bit lower. We put the prolines in the same place. This is an unpublished gel from Nianchuang, just showing that changing these 2 prolines gave nice boosts in expression. So the prolines were at positions 986. In 987, we mutated a cleavage site that separates S1 and S2 and added a trimerization motif. The protein was really stable, looked good by gel filtration. Yeah, and that's the cryo-EM structure, so we were able to quickly determine the cryo-EM structure. We saw in this that there was 1 receptor binding domain in green in the up conformation. The other 2 were in a down state. We have some cool 3D variability analysis to show some of the dynamics as the RBD opens. There's a lot of additional breathing and movements with the NTD. We showed that it could bind ACE2. This was accepted at Science within 2 days with no revisions, and so that's never going to happen again either. But it was good. It was cool to open up our phone and see our structure trending on Reddit. So that was pretty neat. And the paper's been cited over 9,000 times now. So kind of peaked, and it's all downhill from here for publications. What we've been really excited about, of course, is that the 2-proline spike is used in all the different COVID-19 vaccines. So the mRNA 1273 for Moderna uses the 2P mutations, positions 986 and 987, as does Pfizer in BioNTech's vaccine. J&J, working with Dan Baruch and the Ad26 platform, actually did some non-human primate studies where they tested different variants of spike with proline, without. And then the NHP study showed that the double proline was the best one, so that's what J&J used. And then Novavax, which has the purified trimer subunit, also contains the prolines at 986 and 987. So we're always really pleased that something we were working on here at Dartmouth at the bench that maybe was a little underappreciated by at least the editors of some of the journals ended up being widely used. We did want to try to do better, though. So, I mean, that was all done by like February, and we ended up shipping that plasmid to over 100 labs around the world because people were requesting it. They wanted to use it to make spike proteins for diagnostics, ELISAs to figure out who was infected or not, for vaccine development. They wanted to use it as bait to sort B cells and start isolating antibodies for treatment or prophylaxis. And so we did that, and people were just having a hard time making it. Maybe wasn't as stable as we had hoped. The expression was pretty low. So Bill and Melinda Gates Foundation reached out to see if we had a second-gen version. And we didn't, but we were happy to do that. And that was around the time when a lot of labs were shutting down, unless you worked on coronavirus. So some of my colleagues, Ilya and Jennifer, they're like, can we work on coronavirus so our labs don't have to shut down? And we're like, sure. So we decided to do this large structure-based vaccine design effort because now we had the actual structure of SARS-CoV-2 spike. And so Qing Lin and I, my postdoc, we designed 100 individual substitutions, and all of the spikes had to be tested, expressed, cloned, purified, and characterized. And so we needed a lot of manpower to do that. And so the 100 substitutions could roughly be grouped into 4 categories. We had some, 2 prolines work, so we tested additional prolines. Substitutions that would form salt bridges, so in this case, replacing a threonine to a negatively charged aspartate that then interacts with a positively charged arginine. Cavity filling substitutions where we mutate a smaller residue to a larger residue to sort of fill some of these pockets that nature evolved to make it less stable, metastable, and then some disulfide bonds. So it was a huge effort, and all the students and postdocs just grabbed 2 or 5 different variants and cloned and expressed and purified them all. This is expression, so more is better, of some of our top variants. Our base construct was the 2-proline construct. So all of these substitutions were added in addition to the 2-proline. So again, we're getting an expression of around 1 mg per liter. Some of these substitutions, just like glycine to glutamate that then forms a salt bridge, getting like close to 6-fold increase. Several of the additional proline substitutions also looked really good, particularly this one at position 942 gave us almost a 6-fold. Disulfides sort of underperformed, and maybe we just chose bad ones, but those didn't work as well. And we had a couple cavity filling ones that looked okay. And so this work can kind of go on forever. There's no end to the number of variants and combinations, and you can always make a 3rd generation, a 4th generation. So at some point you just have to stop. You just have to like set a goal. And our goal was to try to get something that expressed about 10-fold better and was more stable, easier to work with, and then we would publish and sort of release it to the community. And we were able to achieve that just using additional proline substitution. So here's the 2-proline expression, right? So kind of a fainter band. Combos 14 and 45 had 2 additional prolines, so 4 total. And combo 47 had 4 additional prolines, so 6 total. This is size exclusion chromatography. Our 2p variant is shown here in black. Combo 47, we were getting about a 9 to 10-fold increase in area under the curve. For combo 47, we're also able to shift the melting temperature by about 5 degrees for this first unfolding event. And by negative stain EM, it was just a homogeneous population of prefusion spikes, which was great. Combo 47 didn't sound sexy to us, so we went with Hexapro, which we liked, until we found that there is a protein supplement also called Hexapro. So it is a tasty, unrelated supplement. Hexapro expresses really well at large scale, so we're getting closer to like 11 mg per liter in 293 cells. In XB-CHO, we could get up to 32 mg per liter. Again, a huge increase over just the 2-proline. We're always concerned that our protein engineering may alter structure in some way. So Daniel quickly determined a high-risk cryo-EM structure and showed Hexapro in green, SARS-CoV-2 in white, that the whole S2 subunit or the prolines, they superimpose. Rather than ship these out ourselves, we decided to deposit it at Adgene. They've now distributed it to 215 labs around the world. So the reagent's been used by many. Most of the structures that come out use these, a lot of vaccines and other things. We were curious whether Hexapro is actually just a better immunogen, 1-for-1, compared to 2-proline. So we sent these to Barney's lab, and Olu and Kizzy immunized mice. So sort of a prime boost, 3 weeks apart, and then the terminal bleed at week 6. They tested 3 different doses, 10 micrograms, 2 micrograms, and 0.4 micrograms. And at every dose, Hexapro elicited higher neutralizing antibody titers than did 2P. And particularly noticeable at the lowest dose, where we usually are able to start to tease out some of those differences more. 2-proline spike, sort of a modest immune response, but Hexapro looked really good. There wasn't much of a dose response going from 10 microgram to 0.4 microgram. So we really like it for development as a subunit-based vaccine. It has many companies and academic groups have been using Hexapro to develop vaccines that are in various stages. I'll tell you about 2 that I like. This is for low and middle income countries. So it's a collaboration with Peter Palazzi's group at Mount Sinai. And their vaccine modality was based on Newcastle disease virus. So that's a paramyxovirus like measles, but it infects chickens, avian. It doesn't cause disease in humans. And they inserted the Hexapro spike gene in between the phosphoprotein and the matrix protein genes. And again, because it's an avian paramyxovirus, it can be grown to very high titers in chicken eggs, which is how a lot of the world still makes their yearly flu vaccines. And they can just pivot all of their manufacturing from making flu vaccines to making their own COVID-19 vaccines. So what comes out then is a Newcastle disease virus that's just covered in Hexapro spikes. And then that can be inactivated or not, and some were not inactivated, uses like a live attenuated, immunized, and generate a robust immune response. And so that has been going, a path has been leading that. And we've been working with Thailand, Vietnam, Brazil, and Mexico, among others. And they've finished phase 3. We're waiting on just the final announcement for Thailand. So they call it HXP for Hexapro GPOVAC. It looks like it's going to be ready for use as a booster next year. Mexico calls the vaccine Patria, and it's also approved and ready for use as a booster. And so, again, it can be made very cheaply compared to the mRNA vaccines. They don't have to sort of like wait in line behind other countries to receive their dose, and it gives them their own domestic supply. So we're really pleased about that. One of the other uses of Hexapro that I think is really fascinating is its delivery via a patch. And so this is a patch created by an Australian company called Vaxxus that we have a close relationship with. So what they have is this high-density microarray patch. It contains about 5,000 of these little micro projections. And you coat them with your antigen. And so this is pre-delivery coated, and then post-delivery, and it's sort of from the tip down to the arrow is the amount that's deposited. And the density and orientation of the projections is meant to increase dendritic cell uptake in the skin. So it's you get like 5,000 little deposits of your antigens, so in the epidermis. It's nice because with Hexapro and these spikes, it's stable at room temperature for months. So you don't need cold chain storage, which is always a concern if you're trying to ship vaccines to Africa where they don't have -80 or -20. That is, you could just ship sleeves of these at room temp and it's stable. And it can be self-administered too, which is really nice. So they made this little mini applicator. It's like a tiny tuna can with a spring, and you just put it on your shoulder, push the button, and it applies a consistent force. And so that's nice too, because you could send it somewhere where maybe they don't have a lot of skilled healthcare workers, and people could even self-apply. Preclinical data looked really good in mice. The phase 1 data looked great. And so now they are starting, they're trying to get funding to do the phase 2 studies. And so I think it's another really cool use of Hexapro. It's added stability, and we're excited. And we think it may even have a use not only in low and middle income countries, but even in the US, where some of the vaccine hesitancy is just due to fear of needles, and maybe children would prefer something like this rather than a needle. So that's kind of interesting. Okay, and I will end. When am I supposed to be done?
You're right on time.
Which means I have 10 minutes? I'm supposed to be done? 5? Okay. All right. So we'll do this pretty quick. All right. So I talked about there's a lot of interest in sort of universal coronavirus vaccine, right? Something to protect against the coronaviruses we know of, and as well as those that may spill over in the future. But it is difficult, as I mentioned, right? So just getting one that works against all SARS-CoV-2 is tough. You might try to branch out and get one against all sarbecoviruses, so SARS-1, SARS-2, related ones. You could try to do pan-beta that covers MERS, OC43, HKU1, and then maybe even alpha-2, but it's hard. And there's many different ways of doing it, right? You can imagine some people are doing chimeric spikes, or maybe the N-terminal domain is from MERS, the RBD is from SARS-2, S2 subunits from HKU1, nanoparticles that display many different RBDs. You could just multiplex, and you could just make 6 different spikes from different parts of the tree and inject that, just try to get broad coverage. The one we thought would be most challenging and involve some more engineering was to try to make an antigen that focused on the S2 subunit, which is the most conserved part of the spike proteins, right? So if we look, variables in red, conserved is in blue. So our immune system generally targets the S1 subunit, and the virus then mutates and tries to escape the antibody response. So S1 is highly variable, but S2 is pretty conserved. And this is an approach that's been used by Barney and others for influenza to try to make stalk-directed or headless spike proteins. So that's shown here. So for like flu, HA from Barney's lab and Gary Nabel, they were able to make a headless version. They keep truncating and just try to focus the immune response on the most conserved stem portion. And so we wanted to do something similar. We were working with Romy Amaro's group at UCSD that does molecular dynamics simulations. All right, so this is showing what we're trying to do: pop off S1 and then keep S2 stabilized in that conformation. S2 is decorated with a lot of glycans, which helps shield it from our immune system, because those are self-antigens and we don't raise antibodies against them. So the virus is clever there, really covering this conserved region with glycans. We started with Hexapro S2. Qinglin was able to make some modifications. So that way we just eliminated S1. We sort of started the translation right before S2. We could make the protein, but sort of splayed open like 3 petals just opening up. So we started adding interprotomeric disulfide bonds to help staple that closed. And those work. So this is like a non-reducing gel. We can see the shift to trimers for some of these interprotomeric disulfide bonds, which was working well. Expression looked good. We were able to increase melting temperature for some of them. When we solved the structure, though, we noticed that the apex breathed open. So our interprotomeric disulfide bond was at the base. The apex breathed open. And we could see that by some of our electron microscopy. You can see, like, here's a top-down view of a tightly closed trimer. This one is more open. We sort of had a range of closed to open conformations. We worked with Ralph Barrick's group to immunize animals with either the full Hexapro S2, stabilized stalk that has the interprotomeric disulfide bond, one that lacks the stalk region. And yeah, that's our immunization scheme. Generally, what we saw was that they could protect mice from death with a SARS-CoV-2 challenge. So these mice were challenged with a mouse-adapted SARS-CoV-2. Those immunized with PBS died. And whether we immunized with Hexapro or some of the stabilized stem, it prevented death. Even partially protected against SARS-CoV challenge. They did lose weight, but our stabilized stem in orange actually did about as well as Hexapro. So we were able to get some breadth. So yeah, partial protection, which wasn't bad, but we weren't able to do any better than just immunizing with Hexapro itself, which contains an S2 subunit, which maybe makes sense. We were trying to improve the stability of our S2 only antigen with Romy Amaro's group. And so they were performing weighted ensemble MD simulations. And we're actually able to sort of recapitulate a reasonable trajectory of how the apex of our spikes breathe open. And then this is what it looks like. So you get separation of one of the protomers followed by the other 2 splitting, so top-down view. You kind of see them peeling apart from each other. And so they were able to use these MD simulation trajectories to identify which bonds start to break first, which residues separate. And they were able to design substitutions. It's actually 2 tryptophan substitutions. So here's one, here's a second one. Based on their simulations, this one in particular increased the time until the opening occurred. We were able to make these spike proteins, our base S2, and then if we added one of the tryptophans, the other one, or both, we were able to boost expression. We could shift the melting temperature by about 5 degrees. And this allowed us to then solve a cryo-EM structure of a completely stabilized S2. All the particles were in a closed conformation, which was really nice. But didn't really do anything for immunogenicity. It was basically the same as just the stabilized stem. So that was not the reason we weren't getting robust neutralizing antibodies. So it's an interesting antigen. We're actually using it a lot to isolate antibodies. This was my friend Evelyn Georgiev at Vanderbilt. They were able to pull out an antibody from humans that binds the apex of S2. It binds straight down at the tips of S2, and it binds pretty much every beta coronavirus, SARS, MERS, HKU1. Every single amino acid that it touches is strictly conserved among all beta coronaviruses. It's interesting. It's interesting to think about how it's raised. Like, when does the human immune system see a pre-fusion stabilized S2 that lacks S1? And we aren't sure. It's not particularly neutralizing, though, because S1 is generally there blocking it, but an interesting reagent for us. And we're starting to find other antibodies that bind from the side, and still hopeful for generating some very broadly reactive neutralizing antibodies against S2. All right, so to wrap up, our prior research, primarily almost all of it done here at Dartmouth, allowed for structure-based design of the 2 stabilizing proline substitutions, S2P, that are found in more than 4 COVID-19 vaccines. There's also one in Taiwan, Medigen, and others. Hexapro, which contains the 4 additional prolines. I think it's substantially improved. It's finished phase 3. It's approved in Mexico, waiting for approval in Thailand. The disulfide stabilized S2 only antigens provide partial protection from mouse-adapted SARS-CoV-2 and SARS challenge. The Apex opening, I think, is pretty interesting. I think this is one of the first examples of using MD simulation for structure-based design. And these S2 only immunogens do not appear as promising as the flu-stabilized stems, which is disappointing, but something we felt we had to investigate as a potential option for pan-coronavirus vaccine development. I've tried to highlight many of the people in my lab, some of them Dartmouth grad students like Daniel, collaboration with Andrew for a lot of the early cryo-EM work, Barney, who we've worked with coronaviruses since I moved here with RSV, Jennifer and Ilya's lab, Ralph for some of the S2 work, Romy's group for the MD sim, our funding. Thanks very much, and I guess I can't take questions, so thank you.